Hi, I thought I would do a little overview of the biochemistry of coronaviruses, at least as far as things that I know. So first, to begin, we need a little bit of terminology. The term coronavirus actually is a class of viruses. It's a virus that has a specific morphology. This is pretty detailed, but it's a single-stranded RNA genome. It's inside an envelope. And it's called coronavirus because it's actually derived from the term halo or crown, and that's because of the characteristic crown surrounding the viruses. Um, so under an electron microscope, it appears as if there's a crown around these viruses, and that is why they're called coronaviruses. In fact, many different types of viruses have this crown appearance. These are not a new thing. These have been around for a while, but what's new is the novel coronavirus, or SARS-CoV-2. SARS stands for Severe Acute Respiratory Syndrome. And then CoV is the coronavirus, and 2 is because it's the second variant. The original SARS-CoV virus occurred a couple years ago. It was mainly located in Asia. It spread a little bit, but it didn't cause a global pandemic as we're seeing now. SARS-CoV-2 is thought to be derived from a coronavirus that started in bats. So bats can harbor many coronaviruses because their immune systems aren't developed as much as ours. So the virus just kind of stays there and doesn't cause any problems. And then viruses mutate very rapidly. At some point, maybe in another one of these, I'll talk about why that is, that they mutate so rapidly. So this mutated rapidly enough, and then it's thought it jumped to another animal host. Right now, the thought is that it jumped to this pangolin, which is a scaly anteater type of animal, and then it mutated from there to be able to infect our cells. So SARS-CoV-2 is the actual virus, and then COVID-19 is the disease that's caused by the virus. So coronavirus is just a general term for this class of viruses. SARS-CoV-2 is the current variant that's causing the pandemic, and COVID-19 is the disease that's caused by SARS-CoV-2. I should mention that viruses, by definition, are not living. In another kind of episode of this talk, I'll mention why this isn't considered living, what it cannot do. But sometimes it's easy to talk about it as if it's living. So it's not living. Viruses are not alive. They cannot survive on their own. That's why they need our cells to be able to replicate. And also, when you hear people talking about things, sometimes people mix up these three different terms. But I wanted to just inform you of what those specific terms are. So the structure of the current coronavirus, and this is actually from a picture of other coronaviruses. So it's not all coronaviruses kind of share a similar structure. And so you can see that it has these proteins on the outside of the virus. This is what causes that halo structure. A lot of times you sometimes see these as red, and the red part of this picture is shown as gray. But these outer proteins that are protruding from the surface are called spike proteins. And then this red surface, this is a cutaway view of this other view on the left. This red part is a lipid bilayer, just like our cells have. I'll talk about maybe later why soap works well. Soap is used to disrupt this lipid bilayer. And so we have proteins on the outside that causes the halo. We have the lipid bilayer. And then on the inside, there's an RNA genome. That's what this coily part is. And in the coronavirus, this RNA is coated by another protein, N protein. And then there's a couple other proteins that are involved in holding the structure together and performing different functions. But we have our RNA genome, we have our proteins. All of these proteins are encoded for within the RNA genome. You might recognize the term RNA. This is what I studied in grad school. RNA is similar to DNA. Our cells, as humans, our genome is a DNA genome. So all of our genetic information is stored in our DNA. It then gets converted into RNA through something called transcription. And then RNA gets converted into proteins through something called translation. And in this coronavirus, it kind of cuts out this DNA needing to be transcribed portion. The coronavirus really only works with RNA and proteins. So this is the breakdown of the virus itself, what the genome holds. And so it's just one long piece of RNA. This is all connected together. If you happen to be interested in seeing the sequence of the genome, it has been published. People have sequenced the genome of the coronavirus. And you can see these different regions. These are different types of coronavirus. These different regions make up these different proteins. Again, in another installment, we'll talk about what's missing from this genome that the coronavirus then pulls from host cells. But this is kind of the breakdown. This S portion is the spike protein. That's what's on the outside, again, that causes the halo. So that's what I thought I would talk about for my first little installment here. In the future, I hope to cover some additional coronavirus biochemistry for those who just want a little bit of background on what is going on.